Yeah, give
Sorry,
her a
I wasn't
second.
watching. I was setting up the Zoom. It is a bit, but sometimes this is something that just fixes itself as
Oh, now it's actually better. Yeah.
Yeah.
Okay. Thank you so much.
There may be a few options you can check when you're setting your Zoom to share screen, but other than that, I think we just have to cross our fingers and hope that the Zoom gods smile upon us.
And the internet.
All right, it looks like the YouTube live stream is up and going, so we'll just wait another minute, and Ankita, whenever you want to start, that's up to you.
Yeah, I did put it up on Twitter and stuff.
Okay, great. Thank you for taking care of that. I'm always a bit of a rush before these start, so I'm glad you're on top of that.
Okay. I think we should start. We're like 11:01 and people will start trickling in while I introduce. Okay. Thank you everyone for joining another session of Cell Migration Seminar. And I don't know which seminar this is anymore. We should check that, I guess, at some point. Today we have two wonderful speakers, but first we will go with Dr. Claire Leclech, who got her master's from École Normale Supérieure in Paris and did her PhD in Institut d'Affaires Moulin and Institut Pierre-Gilles Desjeunes in Paris, working on the influence of topological cues on migration of embryonic neurons. And she used a lot of microstructured substrates for that. In 2019, she continued to join the lab of Abdul Barakat in École Polytechnique in Paris as a postdoc, where she's currently working on understanding the mechanisms of contact guidance, response, and physical approaches to collective cell migration. And I guess today that's what she's going to talk about. I will give the stage to you, Claire. Thank you so much for presenting your work. We're really looking forward to it.
Thanks. Let me share my slides.
Jen says we are 70th. You're the 70th.
Oh, I'm glad.
Awesome.
All right. Nope.
There you go. Perfect.
Okay. So thanks a lot for the introduction, and I'm really excited and honored to be able to present my work in these seminars that I really enjoy. And so, yeah, today I will talk to you about some of my postdoc work, mainly focused on the influence of substrate topography on the collective migration on the filial cells. So in our team, we are interested generally in the biomechanics of endothelial cells, which are forming in vivo the vascular endothelium, which is the monolayer lining the inner part of our blood vessels. And of course, it's a cellular monolayer that is really crucial for many aspects of vascular homeostasis. And in vivo, the vascular endothelial cells are subjected to various mechanical and physical cues. For instance, the apical blood flow-derived stresses or the contact-derived stresses. And in my case, I'm particularly interested in one of those physical contact-derived stress, which is the topography of the substrate. Because the vascular endothelial cells are anchored in vivo on a surface, on a vascular basement membrane that is not flat, but that presents a particular topographical organization at different scales, as you can see here on these electron microscopy images. And so the very general question that I'm interested in and trying to answer is to understand how the endothelial cells detect and respond to such basal topographical cues. And to study this question, we are mainly working in vitro, and over the past decades, several kinds of engineered surface have been developed to mimic some aspects of the native topographies. And I put a few examples of them here. And I just want to mention briefly that already during my PhD, I was interested in those questions. And back then I was working with micropillared surfaces. And we've been able to show that actually the shape of this pillar is able to control the morphology and the migration of embryonic neurons. But now for my postdoc, I switched to another type of those microstructured surface, which are microgrooves, and that are very interesting because they constitute controllable and idealized mimics of the anisotropy that is often found in the organization of the extracellular matrices in vivo. And so the main response of many cell types when you put them on nano or micro grooves is actually to align and elongate in the direction of the grooves. And it's a process that has been described for quite some time, and that is called contact guidance. And I put a few examples of different cell types here. But actually the mechanism underlying this particular response of the cell is today not completely understood. So this is typically the kind of question that I'm asking. And while this contact guidance process is mainly observed and studied in vitro, I just want to mention that it's also a relevant phenomenon in vivo, and we have example of this in vivo. I think the most famous example is probably the invasion of cancer cell from the original tumor, and you can see here example of images from intravital microscopy, where you can see that the strands of cells exiting the tumor actually coincide and go along blood vessels or collagen fibers in their environment. But I also wanted to mention a more recent concept and example that is, I thought particularly was relevant in light of the next speaker, which is the fact that it has been observed both in vitro and in vivo that the cells themselves are actually able to secrete and leave behind physical tracts that other cells can after that follow. All right, but so coming back to my original question, so here is the type of substrate that I'm using. So as I mentioned, they are composed of parallel arrays of microgrooves. They are made in PDMS, and this is typically the kind of dimension that we are using. And so after the fabrication of the substrate, we simply coat the surface of the microstructure with an adhesive protein, in our case, fibronectin. And then we simply culture at the surface of the microstructure vascular endothelial cells, in our case, HUVECs. And you can see here that we can observe and analyze the cells at different time of culture and different densities. So here on this time course images of a typical experiment, each element is a cell color-coded with its orientation. And you can appreciate that while the orientation and organization of the cell on a controlled flat surface is quite random, the cells, the vascular endothelial cells on the microgrooves present the typical contact guidance response I was mentioning, where they align and elongate in the direction of the grooves. So around this this question and the system of the microgrooves, I'm working on different projects, but today we'll mainly focus on one of them, on the collective migration of endothelial cells on the microgrooves, and this project was recently published in Nature Communications. So if you want to know more, I invite you to read the paper. Okay, so as I was saying, for this particular project, we were interested in the dynamics of the vascular endothelial cells monolayer on the microgrooves, and it has been shown previously that confining whole cell assemblies, but mainly on adhesive patterns of different shape, is able to generate very specific patterns of collective motion. For instance, here, whole cell rotation on circular adhesive patterns, or bidirectional motion at the edge of rectangular adhesive strips. But nothing was really known about the type of collective motion that we can expect on microgroove substrate. And we really believe it's a different and very interesting setting, because on the microgroove now, each cell is individually constrained by the grooves without any border effect. So to study the dynamics of the vascular monolayers on the microgrooves, we simply record and track the movement of their nuclei for 24 hours and analyze their movements. So you can see here typical examples of the videos that we get. And the first thing that we can notice is that actually, as expected from the elongated cell shape that the cells present on the microgroove, their movement is highly constrained along the groove direction. But now, if we look at a higher scale, for instance, here on this interpolated speed along the groove or on the cumulated cell trajectories, which are here color-coded with the orientation of the displacement vector, we can actually see the emergence of a larger scale pattern of collective motion, which is characterized by the presence of large corridors of cells that are moving in opposite and alternating direction, which we call the anti-parallel cell streams. And I really want to emphasize that the size of this, the width and size of these streams is really higher than both the grooves or the cell size. So we've been able to characterize a little bit further these streams. And first, we've seen that, as expected, the center of the stream is a zone of high cellular coordination, as illustrated here by the red color on this trajectory. And on the contrary, the border, the frontier in between two adjacent streams is a region of high cellular shearing. Then we wanted to test and confirm the collective nature of this pattern of movement. And to do that, we used EDTA to disrupt the formation of cell-cell junction. And in this case, as expected, we lose the coordination in between the cells, but we also prevent the emergence of the streams, as is illustrated here by the dampening in the oscillation that is normally present and represents the streams, showing that the presence of intact cell-cell junctions is really necessary for this particular pattern to emerge. Okay, so then another question we wanted to address was the hypothesis that the streams could be driven by more active kind of leader cells within the monolayer. And to do that, we first looked for cellular heterogeneities within the monolayer, for instance, in cell contractility. And you can see here on this images that actually the organization and intensity of the actin or phosphomyosin light chain staining is actually quite homogeneous in the monolayer, showing that there is no major difference between the cells in the monolayer in terms of cell contractility. We also looked at the cell polarization by analyzing the orientation of the nucleus Golgi vector. And we could observe a homogeneous random polarization of the cells, so without any specific polarization with respect to the grooves, which really shows that actually a sustained polarization of the cell is not necessary for the streams to emerge. So in conclusion, we think that all of the cells are active or at least equally participating in the emergence of this pattern. So then to try and explain the emergence of the streams, we turned to physics and we collaborated with David Gonzalez Rodriguez, who is a theoretician and who built a model that considers the monolayer as an active fluid and the cells as pneumatic particles. And the main specificity of this model is to include the effect of the groove as an energetic constraint on the cell orientation. And this model was actually able to predict a sinusoidal velocity profile across the groove, so much like the one we observed in the experiment. So showing that we are able to successfully predict the emergence of anti-parallel cell streams. And in addition to that, this model also gave us a different prediction. The first one was a linear relationship between the mean cell orientation and the size of the streams. And this prediction we've been able to test and validate it experimentally by changing the dimension of the grooves. We change the amount of constraint on the cell orientation and the cell alignment. And you can see this here. So both on these images or on the quantification of the cell orientation or the cell elongation, we can observe that increasing the groove depth here from 1 to 5 micron is able to increase the amount of cell alignment and elongation in response to the microgrooves. But it also changes the dimension of the streams, as you can see here. So in the case of the deeper grooves, we have higher constraint on cell orientation, but also streams that are longer and thinner, as predicted by the model. And another prediction, or let's say implication of this model, was also the fact that actually what explains the emergence of the streams are not the grooves themselves, but rather the constraint that they impose on the cell orientation. And so if we follow this idea, it means that any source, external source of constraint on the cell orientation should drive the emergence of the antiparallel cell streams. And to test this hypothesis, we subjected the endothelial cells to another known aligning factor, which is laminar apical flow. And you can see that so the cells are aligned in the direction of the flow, and we could also observe the presence of antiparallel cell streams that are very similar to the one that we observed on the microgrooves. So in conclusion, we propose that the antiparallel cell stream can actually emerge in monolayers in which the orientation of the cells is constrained. And to try and generalize this conclusion, we've looked in the literature for other examples of streaming or laning behaviors, which are not that numerous, but in all of the examples that we could find, so either on cellular systems or reconstituted cytoskeleton in vitro, or also purely numerical simulation, this type of behavior is always present where the constituting element of the system are actually externally constrained. So in conclusion, we propose that the mechanism that we have highlighted here in this study could actually be used as a more general framework to explain the emergence of laning or streaming behavior in very different systems. Okay, so the next kind of natural question that arises after that is to wonder about the physiological relevance of such pattern of movements. And for instance, in vivo in the blood vessels, we know that the vascular endothelial cells are aligned and elongated in the direction of the blood flow. So we can think that in theory, the streams could be observed and present in vivo, but we're really missing actually characterization of the dynamics of the cells in vivo. So this is hard to check. But I want to end by coming back again to my first example of the strands of cancer cells invading the tissues. And I hope that you can see here on this video actually the presence of antiparallel movement, which suggests that this type of pattern of motion can also be present in vivo. Okay, and actually in the last part of this talk, I want now to advertise about a new project that we've been developing in the lab, still using these microgrooves. But now let's switch scales and look more precisely at the dynamics of the nucleus on the microgrooves. And you can see here on this video that actually, when we really look more precisely at the nucleus, we can see that they are undergoing major and dynamic changes in their shape with the presence of these phases of movement where the nuclei are highly elongated. And when we looked more precisely on 3D reconstruction of the nuclei, we could actually observe that the microgrooves are able to significantly deform the cells and their nuclei. And this very elongated nuclei that I was mentioning before actually corresponds to nuclei that are completely trapped and caged within a groove. And actually, we could identify in our microgroove system different configuration of nuclear deformation. And we believe that it's a very interesting observation because, as you know, nuclear deformation and nuclear mechanics are highly studied today because of their involvement in different pathologies, among others. But usually the nuclear deformation are studied in experimental systems such as microfluidic systems that are experimentally still a bit complex and low throughput. And we believe that with our microgrooves, we have a new, much simpler and high throughput system to induce and study nuclear deformation. So this is something that we are really developing today. And to mention some of the results that we have so far on this. So first, we were very interested in understanding the links in between the nuclear deformation and the nuclear mechanics. And to do that, we performed Afm microscopy. And so we probed the stiffness of the region above the nucleus, which we considered as a good approximation of the nuclear stiffness itself. And by doing that, so first we've been able to measure very generally that the nuclei on the grooves are softer than on a flat surface, which can either be a cause or a consequence of the fact that the cells are more deformable on the grooves. But now, maybe more interestingly, we also probe and followed the stiffness of a single nuclei in time undergoing caging or uncaging transition. And we've been able to show that both of these transitions, so either going in or out of the groove, is associated with a transient softening of the nuclei. So we believe that the cycle of the dynamic cycle of deformation of this nuclei are actually associated with a cycle of changes in the nuclear stiffness, which also confirms the link in between the nuclear deformation and their mechanical properties. And to finish still on this idea of nuclear deformation and mechanics, we have also been able to test in our microgrooves the behavior of cells with a mutation in lamin AC, so the major component of the nuclear envelope. And we've been able to measure and see that these nuclei that are mutated for lamin AC are more deformable in our microgrooves. And we really see the emergence of a very particular and aberrant configuration of deformation, which really shows that these nuclei are more fragile and which confirms once again the link in between the nuclear deformations on the microgrooves and their mechanical properties. And I will stop here and conclude on this presentation. So I really hope today that I convinced you of the potential and the interest of this microstructured and microgrooved system. So we are using microgrooves here as an in vitro tool to mimic extracellular matrix topography. I've shown you that we can use microgrooves as a tool to control collective migration patterns, but we can also use it as a tool to control cell shape, monolayer organization, as well as now a tool to study nuclear deformation and mechanics. And so in conclusion, we really think that this constitutes a powerful in vitro tool to control and understand various aspects of cell biology, from cell migration to cell shape regulation or even cell biomechanics. And I'll stop here by thanking all of the people involved in this work. So the team of Abdul Barakat, all of the collaborators, of course, our funding, and you for your attention, and I'll be happy to take questions.
Thank you so much for such a wonderful talk, Claire. We already have a few questions. The first question is from Anna asking, did you look at the orientation of focal adhesions, and did you test the cell alignment and elongation in absence of focal adhesions, like plating the cells on polylysine?
Well, that's actually a very good question, since we have actually just done that.
Amazing.
So yeah, we've been studying the morphology of the focal adhesions on the grooves and on different groove depths. And so we've been able to see, as previous studies, that the focal adhesions are aligning in the direction of the groove, and they're especially localizing at the edge of the region. And they are even more aligned and elongated on the deeper groove, so where the cells are even more aligned and elongated. So they seem to be responsive to the topography. But now, surprisingly, if you actually use the polylysine coating to allow a weak interaction with the substrate without the formation of focal adhesions, you retain the same type of contact guidance, and you even retain the difference in between the groove depths, meaning that even without focal adhesions, the cells are still able to respond to the topography. And so then we've done some other experiments that actually show that in this case, it's more the cell protrusions that seems to be like the major initial player in the detection of the topography. But probably in vivo, they kind of cooperate together, but the focal adhesions are not alone necessary for the contact guidance.
Okay, fantastic. Thank you. Before I go on to the next question, I just want to remind everyone that if you want to ask your question yourself, just type question in the chat or raise your hand and we'll call on you. And that goes for folks who've already put their questions in the chat in case you want to say it yourself instead of hearing me and Ankita talk too much. All right, the next question is from Emily Mace. They're asking, as a follow-up to the previous question, Anna's question, have you considered whether differential integrin usage could be involved? And they're thinking about work from Dan Hammer and Jan Burkhart showing that T cells use different integrins under shear flow to go upstream versus downstream.
Yeah, so that's a good suggestion. So no, we have not gone that far in the focal adhesion study. So we mainly did Paxillin staining, like the typical staining to look at focal adhesions. But yeah, that's definitely a good idea, and we can look more this way. Thanks. Yeah.
Thank you. Absolutely amazing. Next question is from Noah Michel asking, given the strong nuclear and cellular deformations.
Actually, he can ask his own question. He put his hand up. Yeah, did as instructed. Yeah. Thanks, Noah. Go ahead.
Yeah. So that was amazing. Wonderful talk. I'm just curious. So given you have these extraordinarily strong nuclear deformations and probably like the cells are so deformed too, do you think the Golgi orientation with respect to that highly deformed nucleus is kind of like maintained so that that's still a useful vector to define the direction of motion of the cell?
Yeah, that's actually a good point. Because it's true that on the polarization analysis that I showed, where we see no specific polarization, we did not distinguish in between the different type of nuclear deformations. What I can say is that it's true that when you look specifically at the cage nuclei, so the ones that are completely within the grooves, in this case, the Golgi seems to be also a bit trapped either in the front or in the back of the cell. But we have not gone into further detail on that, but I think I will keep an eye for it. That's a good suggestion.
Thanks.
Amazing. Next question is from Kalina, if she wants to ask herself. But do you have any hypothesis as to why you see a softening of the nucleus when caging or decaging? Also says it's a wonderful talk.
Yes, so no, we are very, it was a very interesting observation for us as well, but we don't have any idea so far on the underlying mechanism. So yeah, I cannot really say more about that, but we definitely need to look more into this direction.
Okay, great. From Amkar Joshi, saying thanks for the great talk about the nuclear deformation. Are there any particular triggers that drive the cells towards particular nuclear deformations? And does this influence their movement speed or anything else you've observed?
So on this, what we've shown and measured is that we are able to control the type of deformation that we induce on the nuclei by changing also, once again, the groove dimension. So for instance, if we increase the groove width and spacing or the groove depth, we favor the presence of this fully caged nuclei at the expense of we have nuclei that are deformed on several grooves and ridges. So we are able to control, and that's why also the system is interesting. We can really kind of control the type of deformation we are inducing. And then concerning the link with the speed, what we've been able to measure is that when the cells and their nuclei are in the cage phase, this corresponds to higher speed of migration, usually. Probably involving, contractility, because in these phases they are a bit more confined, so that's probably why, but this we've been able to measure, yes.
Amazing. Next question is from Srinivas, asking, have you looked at or do you plan on looking at ERK wave in these anti-parallel band of cells?
So yes, actually, so now the idea that we have to kind of bring this project forward is that, so we think that with the grooves, we have a good system to really control the cell shape, right? And especially in monolayers, which is not that studied. And so now we want to use this system to study and address the consequence of having aligned monolayers on functional endpoints. And we are interested in looking at the directionality of collective waves, so such as calcium waves or ERK. So yeah, we will definitely do that. And well, I think we will have some surprises. I don't know what to expect in terms of the anti-parallel cell streams, but maybe we'll be able to see something related to that.
Okay, great. I think we have for our last question from Stefano, who will be speaking later in the series. So great to hear from you. Oh, wait, one more from Anna. Okay, so Stay's asking, what's your prediction for zigzag grooves, and would the cells have perhaps less coordinated behavior?
So that's always I always get questions on, whether to test this or this type of geometry of grooves. So on zigzag grooves, so what what we have shown in this study and what the model is telling us is that normally if you orient your cell by any cue, you should be able to generate typically anti-parallel cell streams. So if I'd say the wavelength of the zigzag or allow still the orientation and alignment of the cell, we should expect to see a similar behavior. But then it's true that that probably with by disrupting a bit the shape of the cell, you can also disrupt the cell-cell junction and prevent some of the coordination and collective behavior. So this is true, and I guess it would depend maybe on the dimension of the zigzag. And also one idea that we have, but that we didn't test yet, was to use circular and concentric grooves to see also if we could kind of generate anti-parallel rotation in this situation, but we have not tested that yet.
Okay, thank you so much. I'm actually, just in the interest of time, I'm gonna have us move on. You do have one more question in the chat, Claire. Maybe you can.
I can answer on the chat.
Great. Thank you again so much. Thanks for that fantastic presentation. I have a question afterwards if we have time as well.
Okay, no worries.
All right. Francesco, if you want to go ahead and start sharing your screen while I introduce you. So today we are lucky to have two researchers presenting their work. And so I'm delighted to next introduce Dr. Francesco Bashiri. He got his master's in biology from the University of Pisa in Italy. And for his PhD, he joined the newly formed lab of Hesso Ferran at the University of Constance in Germany, where he worked on the role of CDC42 in cell polarity. Francesco then moved to the Gustave Roussy Institute in Paris to start his postdoc in the lab of Guillaume Montauk, where he focused on mechanobiology and clathrin-mediated endocytosis in cancer. And now, in his position as a researcher, he's working on understanding how cancer-associated fibroblasts contribute to shaping the tumor microenvironment by favoring migration of other cell types. And I believe that is the exciting work that we're going to hear about today. So with that, Francesco, the floor is yours. Thank you so much.
Thank you, Jennifer, for the introduction. And thank you also for selecting me to present at this great series of seminars. I think we can all agree that these seminars are one of the very few good things that came out from COVID. As Jennifer said, I work in Gustave Roussy, which is a cancer hospital. So we are very interested in cancer. And what we used to say is that cancer is a disease where physics matters a lot. In fact, during the progression of the disease, you have a lot of physical parameters that change. For example, you have the extracellular matrix that gets stiffer with time. You have cells overgrown in confined space, which induces a compression. And finally, you also have cells that escape the primary tumor and find themselves in a completely new environment with a totally different architecture, where actually the model that showed us can turn out useful to understand what happens at in this place. And indeed, what we do in the lab, we are interested in this very last step of the disease, metastatic dissemination. And as the metastatic dissemination, as all the other physical modifications that occur during cancer, is associated with fibroblasts. Fibroblasts are non-transformed cells that are present in the extracellular matrix. And usually, fibroblasts are responsible for maintaining the architecture of tissues. However, what happens in cancers is that fibroblasts become hyperactivated. So we start to talk about cancer-associated fibroblasts, starting with CAFs. And what it means that they become hyperactivated, it means that basically they highly remodel the extracellular matrix. They cross-link collagen fibers, making the matrix stiffer. They secrete fibronectin, collagen, and many other proteins. They pull the already existing fibers, aligning them. And all of this contributes to the progression of the disease. So by looking at the last step of the disease, metastatic dissemination, there are a lot of open questions in this field because so basically we don't know anything about how metastatic cells disseminate away from the tumor. We know very little about this. And as biologists, when we started to think about, many years ago, people started to think about how cells escape the primary tumor, the first answer that came to the mind of biologists was chemotactic gradients. So a chemotactic gradient probably drives the escape of the cell from the primary tumor all the way to the blood vessels, where the cell then will enter the blood vessel and disseminate away to generate distant metastases. However, there is very little evidence for the existence of chemotactic gradients in vivo, and instead, what was observed and measured is, again, physical changes. For example, what the Weaver lab has seen is that in breast cancer, you have cells escaping the primary tumor there where the extracellular matrix is stiffer, like if there were local rigidity gradients. So you have stiffening of the extracellular matrix that can influence the process of metastatic dissemination. And we just saw it in the talk of Claire. Aligned collagen fibers are a very bad prognostic sign because cells interpret these aligned fibers as a migration highway. So they start to migrate far away from the tumor. And these two parameters are controlled by CAFs, cancer-associated fibroblasts. So in the lab, we decided to take a look at CAFs. We took some CAFs from tumors and we embedded them in a 3D network of collagen fibers. Here you have the collagen fibers in green, and you see the CAF marked with a plasma membrane marker in magenta. And what we observed was something a bit unexpected. We saw that the CAF was leaving behind a lot of material marking a migration path. So basically, there were some tracks forming behind the CAF. And this behavior was curious. Also, it was very prominent, so the gel was full of these tracks. And this prompted me to investigate the behavior more closely. So I went to 2D, and in 2D, what I saw is that virtually all migrating CAFs leave behind some tracks. And these tracks seem adherent to the substrate. And in fact, you can see them by interference reflection microscopy. This is a technique that allows you to see what is bound to the glass in this case without the need of any specific fluorophore. And you can see only the things that are really sticking to the glass. So you see the tracks are really adherent. And not only they are adherent, but you can visualize them also by pulsing, which is a cytoplasmic marker. So this means that tracks are tubules, are lipid tubules that are closed, that contain some cytoplasm and probably also other materials from the cell of origin. Because tracks are seem to be adherent, We also thought that probably they were containing some integrins, which are the adhesion receptors of cells. And in fact, what we observed is that tracts are full of integrin beta-5 and of its partner integrin alpha-V. Instead, other integrins like integrin beta-1 or also integrin beta-3 that I don't show you here, they remain in the cell and they are not left behind during migration. And also, if you deplete integrin beta-5, you completely lose the tract. Especially in this picture, you can see that the tract has left behind a very long tract. This tract is 0.5 millimeter long, which from the point of view of a cell, it's very long. And this made me question about the stability of tracts. Are tracts very stable over time once they are deposited? To test this question, I plated some cuffs on gridded Kolbe slips. I then removed the cuffs once they have deposited the tracts and monitored tracts over time, observing that even after 3 days from deposition, tracts were still present. So basically, almost 80% of tracts were still present after 3 days. So tracts are very stable features of the microenvironment. So now you're probably all thinking about microsomes, because this is something that recently came up in the literature. We all know about the papers from the EU lab. And in fact, we see microsomes. So this is a tract. It's a uniform tract. Here you have the cell, and the tract is still connected to the cells. And we can see some microsomes forming on the tract. Now I will show you what happens when the cell leaves the tract, when the tract is severed from the cell. Here is the video. I replay it one more time. You see, as soon as the cell leaves the tract on the ground, the microsomes disappear. So as described in the original paper of microsomes probably burst, releasing their content. And this occurs in a time span of 2 hours and 35. So microsomes are very short-lived, whereas tracts, they stay for several days on the substrate. In addition to this, because we knew that integrin beta-5 is sensitive to the stiffness of the substrate, we tested whether we could affect tract deposition by modulating the stiffness of the substrate. And indeed, we saw that cuffs leave more and more tracts when we increase the rigidity of the substrate where they migrate. And this implies that wherever you have a stiffness gradient, you will also end up with a tract gradient. Of course, in 3D, things might will for sure be more complex, but at least in 3D, this is the case. And this parallel between a stiffness gradient and tract gradient was something interesting that prompted us to investigate whether tracts could affect also the adhesion or the migration of cancer cells. The first thing I did was to generate substrates conditioned by the presence of tracts and to let cancer cells adhere on these substrates. Then I counted how many cells will adhere on tracts versus how many will adhere outside. And you can see that the ratio is strongly in favor of cells adhering on tracts. And this holds true even if all the substrate is coated with collagen 1 or fibronectin, meaning that the tracts are very potent adhesion fuse core cells. So I tried to do some fibronectin stainings, and we could not see fibronectin below the tracts. However, we could not be sure that the tracts were deposited alongside some other proteins of the extracellular matrix. To be sure that the cells were really recognizing the tracts and not something else, what we did was to touch the tracts with an integrin beta-5 inhibitor, CyNMG type, which would leave unaltered all the extracellular matrix eventually deposited along the tracts. And again, in this case, we see that the cancer cells, they completely lose the ability to preferentially accumulate where the tracts were deposited, meaning that now, in the absence of tracts, cancer cells adhere equally well everywhere. So the question was, how do cancer cells adhere on tracts? How do the cancer cells see the tracts? And we tested whether this was focal adhesion mediated, and we could not see any focal adhesion forming on the tract. Here you see we used the vinculin as a marker for focal adhesions, and most importantly, we also depleted cadmium 1 essential component of focal adhesions, and this did not inhibit the ability of cancer cells to find the tract. Instead, what we saw is that AP2, a protein known for its role in endocytosis, was strongly accumulating along tracts, and its depletion was reducing the ability of cells to migrate on tracts. Instead, clathrin, which is, as the name suggests, a main component of clathrin-mediated endocytosis, clathrin depletion did not alter the ability of cells to find tracts. So what is clathrin-mediated
Francesco,
endocytosis?
can I just interject real fast? A couple of folks are saying that they're having a little bit of audio trouble hearing you. I'm not sure if it's anything that you can fix on your end. Just a little bit of patchiness.
Can you hear me now?
I can hear you just fine. Let's go ahead and see if that helps. Thanks so much.
Something changed?
Not sure.
It sounds good to me. It's just sometimes it's a bit patchy. Somebody is suggesting to, yeah, we can hear you.
So is somebody here?
Yes.
I can't hear anything.
Oh,
yeah, go ahead. I think this sounds pretty good.
Okay, if it's okay, great. Sorry, I put an external microscope, my microphone. So, very briefly, just to mention how clathrin-mediated endocytosis works. Clathrin-mediated endocytosis is a process that, of course, oh, no, it doesn't work anymore. Okay, clathrin-mediated endocytosis occurs at punctuated spots of the plasma membrane, known as clathrin-coated structures or clathrin-coated pits that are nicely visible under the electron microscope. It's these ones that you see, these domes that you see in the picture on the right. And the structure you see, the honeycomb shape, this is clathrin. So basically, when you have a receptor that gets activated by binding to its ligand, this induces the recruitment of adapter proteins like AP2, clathrin, and this newly formed coat drives invagination of the membrane that leads to finally to the generation of a vesicle that will leave the plasma membrane, losing its coat and terminating the process of endocytosis. What we have observed in Guillaume lab is that, so usually this process is very fast. This is known. It's like it takes approximately 1 minute to complete the internalization of cargo. But in Guillaume's lab, we have observed that there are several ways by which you can frustrate this process and making it last for several hours. For example, one of such ways is to let the cell try to internalize an oversized cargo. So usually clathrin-coated structures are domes of 100 nanometers of diameter. And what happens if the cell tries to internalize a cylinder is that these domes cannot form. And what happens is that the domes become galleries. You can see in this picture, this is a paper we published a few years ago, and you see the clathrin forming around a collagen fiber. Not only the shape changes, but the structure is frustrated. Endocytosis cannot be completed. These structures are very long-lived, and basically they become atypical adhesion structures that grab the collagen fiber. So we thought that the same was happening along cuff tracts, and in fact we could see by fluorescence microscopy that clathrin-coated structures forming along tracts were long-lived. And we confirmed that actually these structures were grabbing the tracts by super resolution microscopy. Here you see the tract, and you see AP2 structures forming on top of the tract. And what you can see also is that the clathrin structures seem to be able to extract some material from the tracts. So these are the differences that we observe between tracts and collagen fibers. The first difference is that you can internalize portion of the tracts, and the second difference is that the tracts are adhesive, they are adherent to the substrate. So basically, probably they oppose a resistance to the internalization. And to win this resistance, the clathrin-coated structures recruit locally actin that will contribute to the termination of endocytosis. To test whether this was true, I did this very simple experiment. Here you see on the left, cancer cells adhering along tracks, and you see that the track disappears very quickly in a matter of hours. And you see a lot of endosomes forming, implying that the cells have internalized the tracks. If I let the cells adhere in the presence of cytokalasin D instead, the cells find the tracks, the clathrin-coated structures form along the tracks, but the tracks stay except from some bleaching that is due to the lasers of the microscope. And you see here there is a strong frustration. Just to tell you how strong this frustration is, the structures that are supposed to be 60 seconds long, now they last for 12 hours. So it seems that we are dealing with frustrated endocytosis, and that actin plays a role in internalization of the tracks. We also did some mass spectrometry on the tracks to try to identify the receptor that allows clathrin-coated structures to form on the tracks. And we found that these two proteins that contain RGD domains are involved in the recognition of tracks. So basically the model we have is this one. You have the track that is immobilized to the substrate by integrin alpha V beta 5. You have the cancer cell arriving on the track and recognizing the track by the mean of integrin alpha V beta 3 contained in clathrin-coated structures. Here, there is a video to see where we will validate this model. Here there is a cancer cell. In yellow, you see the integrin beta 3, and in green, you see clathrin-coated structures. You see, so as soon as the cell arrives on the track, you see accumulation of integrin beta 3 on the track. And after a while, integrin beta 3 is gone and there is only AP2. We were a bit surprised by this because we thought that integrin beta 3 would stay, but probably integrin beta 3 is only needed at the beginning to stabilize the curvature, and you don't need it anymore after a while. And in fact, if you block endocytosis by cytokalasin D, then you see accumulation of integrin beta 3 and AP2 at the same time. So very, very quickly, the last part of the talk, you probably saw it also in the video before, the cancer cells not only adhere on the tracks, but they can migrate on them. And it seemed that the cancer cell could migrate directionally on the tracks, always in the same direction, meaning the direction in which the calf had migrated. To test whether this was true, we developed an assay based on durotaxis. So we generated rigidity gradients, and we measured by manual tracking of the cells their ability to migrate towards the stiff part of the gradient. So here you see the two cancer cell lines, breast cancer cell line MDA MB231 or colon cancer cell line HCT 116. They have a forward migration index of 0. That means that they migrate on the gel, but they don't have a preference towards either stiff or soft. They randomly migrate. And cuffs, instead, they migrate strongly towards the stiff. So what we did was to plate cuffs on the rigidity gradient. We let them generate tracks. Then we put cancer cells on the gradient, and we let them migrate in the presence of tracks. And at this point, the cancer cells were becoming durotactic. As a control, we took the same gradient. In addition to removing the cuffs, we removed also the tracks. And in this case, the cancer cells were migrating randomly again. So to sum up, cancer cells migrate randomly, cuffs are strongly durotactic, cancer cells on cuff tracks become durotactic, and if we remove the tracks, the cancer cells migrate randomly again. This is due to AP2 structures forming on tracks, because if we put cancer cells depleted for AP2, the cancer cells migrate randomly, but in agreement with the data I showed you before about adhesion along tracks, clathrin depletion does not affect the ability of cancer cells to follow the tracks and restore durotaxis. And in fact, there is some data in the literature showing that if you induce the curvature of the plasma membrane by other means, for example, by adhering along a track, you just need AP2 to form these endocytic structures, and you don't need clathrin anymore. So to sum up what I showed you that fibroblasts can produce tubular tracks that become very stable features of the microenvironment, that these tracks can be recognized by other cells by frustrated clathrin-coated structures, and the tracks can drive migration in a polarized fashion. So with this, I'm done, and I want to thank, of course, Guillaume, my supervisor, and all the lab for help and constructive discussions, and all the collaborators, especially Sandrine and Abigail, who helped us a lot with super resolution microscopy. And I thank you for listening, and I'm ready for questions.
Thank you so much. That was really just beautiful work and fantastically interesting. I'm going to use my privilege here and ask a couple questions first. Sorry, y'all. So I have two questions. First, in terms of the mechanism of deposition from the calves leading these tracks behind, do you think that it's intentional, cutting some, a deposition process, or do you think it's just ripping off pieces of the cell that are just, they're adhered, cell needs to move on and it leaves it behind? I know you can't really assign intentionality to the cells, but that's kind of what I'm getting at.
So it's a question I find fascinating because it has a lot of implications. I think that for some reasons, integrin beta 5 is not reabsorbed by the cells. So there is the track is severed. It's not, there is no secretion like exosomes. It's more passive in this way, but it's so much material that it must fulfill a function. This is my opinion. So I don't know if I answered completely your question.
No, it's interesting because there's older papers where they sort of talk about ripping off bits of cells and leaving them behind. And I think they use that language. But of course, there isn't a lot of follow-up. And so, yeah, I was wondering what you were thinking. And then my second question is, in terms of the cancer cells that are following the tracks, do you think that there's any role, it's sort of along the lines of Claire's talk, is there a role for just the topography of that, there's a physical shape being left behind, or do you think it's more of a biological cue that they're following, biological breadcrumbs?
So I think that in our system, in the rigidity gradients that we generated, what the cells follow is topography. In vivo, it will be more complex. So I would say that in vivo, the tracks would represent some sort of signs that the cells find on their migration path. So probably the main topographical role is still left to the collagen fibers in vivo.
Amazing. We'll take the questions from our group here. First question is from Anna, says, very interesting talk, and I agree. Do you call tracks to these retraction fibers where microsomes were reported to be located? Does the length of the retraction fibers are dependent on contractility and or persistent migration?
Yeah, these are great questions. I don't have answers to all of them. So the relationship between, so these tracks, they are probably retraction fibers. They are not only retraction fibers because, for example, MDA and B231, they have a lot of retraction fibers, but they don't leave tracks. So it must be a subset of retraction fibers. I'm not fully able to explain this point because so I don't know more than this. I think the tracks are just the retraction fibers that contain integrin beta 5 for some reason. The microsomes instead are present on all retraction fibers. In fact, cells that don't leave tracks, they can leave microsomes. And the length of the retraction fibers, whether this depends on contractility or persistent migration, there is a paper that came out last year showing that when cells turn, the type of tracks they leave changes. So yes, definitely there is a correlation between the shape of the track and the type of migration, but it's not clear exactly how this works. So one thing is, for example, the angle at tracks, because tracks you saw, they are very branched. So the angle in the case of calves is always the same, is approximately 60 degrees. And probably with other cells, it will be a different angle.
Okay, great. Thank you so much. Elizabeth, do you want to ask your question next?
Yeah, thanks for the great talk. I really enjoyed it. My question really focuses on the like cancer specific aspect of this. So are there other fibroblasts that have been seen to deposit similar tracks, and are cancer cells the only ones that can follow these tracks? Or do you think if you put, you know, an immune cell or like epithelial cell, if they would cancel, and could it be kind of a like in cancer, you know, attracting these normal cell types towards tumors?
Yeah, thanks for these great questions. As for the cuffs, for what I can see now, it doesn't seem to be specific for one subtype of cuffs. I took cuffs from different types of tumors, and probably they were mixed populations of cuffs, and all of them were leaving tracks. But I have to say I have a very little amount of samples. So for the moment, it's just less than 10 patients. So I don't know. As for which kind of cells can follow the tracks, tracks can be followed also by healthy cells, not only by cancer cells. Cuffs themselves, they can follow the same tracks, the tracks they leave. It's just that if a cuff follows the track, then the tracks become reinforced because there is more deposition. And if a cancer cell follows, the track is eaten, so it disappears. And for immune cells, I don't know. I never tried, but I would love to test this possibility.
That is so cool. Next question is from Srinivas asking, have you looked at the traction force applied by these fibroblasts on the ECM? And does the force coming from actomyosin affect the rate of deposition?
These are great questions, and I didn't look at forces. I would love to do this, especially because a few years ago, we published a paper about what is now called reticular adhesions. It's a specific type of adhesion that is integrin beta 5 based, and it's not yet clear whether reticular adhesions can generate forces, if the forces are in the same order of magnitude of focal adhesions. So this would be extremely interesting to do.
Okay, great. Claire, since you're do you want to just ask your question?
Yeah, I can.
Sorry to call you out, but
no, that's okay. So I was just curious and wondering if I understood well, these tracks are more like pieces of the plasma membrane that is left behind, right? But have you looked also at the secretion of ECM proteins by the calves? Because, for instance, we know that in our system, the cells secrete and deposit some extracellular matrix, and could they be kind of related to the tracks you're looking at in terms of, I don't know, a template for the deposition of the tracks or the other way around?
So actually, with the cuffs I'm using, I used in the pictures I showed you, they come from Daniela Vinjevich's lab, and they see that these cuffs secrete a lot of fibronectin. So fibronectin was the first thing we tested, but I could not find any fibronectin below the tracks or next to the tracks. Actually, I could find very little fibronectin deposited by the cuffs.
Okay.
The only explanation we have is that we use cuffs at very low densities. So probably there is a density switch between a secretory and non-secretory cuff.
Okay, yeah, thanks.
Awesome. Francesco, are you okay sticking around? There are a few more questions.
Yeah.
Okay. Next question is from Adriana asking, do you find cross-linking enzymes in these tracks?
I didn't find any locks. I don't know about other enzymes. I should check, but that would be very interesting. Thank you for
I feel like we are writing your future work here.
Thank you. It's very helpful.
Great. I can take the next question, Jen. Srinivas Alanki is asking, do normal fibroblasts also deposit tracks as well? And what about the other cell types like immune cells? Do they deposit as well?
Yeah, so I love this question. Thank you for asking. For normal fibroblasts, the problem is to define normal, because well, tracks are secreted, are let's say, are seeded only on stiff substrates. and rigidity is a way to activate cuffs, to activate fibroblasts. So even if you take normal fibroblasts, which we did, we have fibroblasts that were taken from a biopsy away from the tumor. These are supposed to be normal. They leave tracks on stiff. We tried osteoblasts, which are very specialized fibroblasts. Let's say they leave tracks as well. As for immune cells, I don't know. So actually, there is evidence that macrophages leave tracks, but I didn't test it myself.
Okay, thank you. That's super interesting. From Felix, saying very nice work. Any idea how different integrins are sorted in or out of the tracks? And has anyone done any proteomics of the structures?
Yeah, thank you, Felix, for this question. No idea, but I want to do this. I hope I will be able to answer you in a couple of years. If somebody has ideas, I will be happy to share experiments.
That's amazing. Next question is from Tae-Yung asking, from one of your data about the MDA cells, they do not show durotaxis when the tracks were washed off. So he's wondering, do you think the previously known durotaxis is actually from these tracks?
We thought about this possibility. And for example, what I tried to do was to seed cuffs at very low densities on these durotaxis gradients. And in fact, like for example, if I put only 100 cuffs, I still see durotaxis, but it's much less than when I put 10,000 cuffs. So it might be that tracks contribute to improving the durotaxis, but still I think that there must be something else that gives durotaxis to the cells.
Yeah, it's really interesting because, you know, so many people report durotaxis and then other people report, oh, no, it doesn't happen. And then other people say it happens in the other direction. And so we've got a lot to figure out there. From Metello Innocenti says, long time no see. I guess you two know each other. And is wondering, do you see these membrane tracks in 3D matrices? And also in the past, again, they found that normal fibroblasts leave behind tracks. And so he's wondering about specificity.
So yes, we see tracks. I see tracks in 3D collagen networks. I see tracks in decellularized organs. So matrix that I obtained from organs, the mouse organs that I decellularized. And yes, normal fibroblasts, we come back to the question of a couple of minutes ago. It's difficult to define normal fibroblasts because tracks are rigidity dependent, and when you put cuffs on, when you put normal fibroblasts on a rigid substrate, you might activate them to myofibroblasts.
Awesome. I think this is last question in the chat unless somebody else asks one. The question is from Clotilde Calderwood asking, do you think the type of integrin present in the tracks depend on the matrix the calf is migrating on? Because she has seen beta-1 integrin positive tracks left by fibroblasts cultured on fibronectin.
That's a possibility. Actually, I tried on fibronectin and I couldn't see integrin beta-1 tracks. I still saw beta-5 tracks. The fact is that beta-5 is the receptor for vitronectin, and you have a lot of vitronectin in the serum. So we constantly see vitronectin in all our mass spectrometry. So I think there might be problems with this. So probably we always see beta-5 because even if we could be fibronectin, we still have vitronectin as well.
I have a question actually, which is going to be very biased on the stuff I'm interested in, is thinking about cells which do not really depend on adhesion-based migration. What do you think about like anything like blood-based migration or something would do with these tracks?
So something that I find interesting, for example, is this experiment I did with cytokalasin D, where basically the cells have really problems to adhere. They remain round. And when they find the tracks, they are not round anymore. They are not completely adherent, but they sort of spread. And I think that something similar could happen also for not very adherent cells. It could be a way to force adhesion, or to, for example, to induce rolling and adhesion on blood vessels to extravasate. That's just pure speculation.
That's cool. Anybody else would like a question? I think we are done with the chat thing, right? Okay, go Jen.
No, great. Thank you both so much, Claire, Francesco. Those were really fantastic presentations. It's so exciting to see what you guys are working on. Is that work published, Francesco? Because I know Claire's, most of it was.
We just put it on bioRxiv. So yes,
okay, on bioRxiv.
It's bioRxiv.
Great, awesome. Well, thank you both so much. That's really, we're really glad to have you both, and wish we were in Paris with you. And with that, I'm gonna sign off from the live stream.